Okay, this is the fourth seminar, Bioengineering 599J in spring quarter. And we have an external speaker today. His name is Dr. Abraham Lee. He's a program manager in DARPA. And also, this is his first week as a senior technology advisor to NIH. And Dr. Lee got his BS degree from National Tsinghua University in Taiwan, and master's and PhD degree from UC Berkeley in 1989 and 1992, respectively. And then from 92 to 98, he worked at Lawrence Livermore National Laboratory Center for brand new microtechnology at that time. In the beginning as a postdoctoral fellow, later principal research engineer, and then group leader. Since the February of 1999, he has been the program manager in DARPA, the Defense Advanced Research Project Agency, managing the program Microfluidic Molecular Systems. And then since the April of this year, he's transitioning into NIH, and he told me this was the first week working at NIH, still having responsibility at DARPA. Dr. Lee will give us a presentation today. He has a number of publications, and also he told me the number of patents increased by several in the last few months. He has 20 patents. And today he will give us a presentation with the title of Microactuator for Biomedical and Biotechnological Applications. Hey.
Here at the Pacific Northwest in this beautiful campus, and glad that there's no rain and there's no earthquake yet, so I will hold our breath. Well, again, thank you very much for inviting me. Today I'm going to talk about microactuators for biomedical and biotechnological applications. As Dr. Kim mentioned, I just started this week in National Cancer Institute, so maybe perhaps throughout the talk I'll show you a little bit of how some of my plans of what I'll do there in the next few months between the two agencies, and why there is a need for someone to be a liaison between the two agencies. I'm going to divide my talk into three main categories. The first is I'll spend 3 to 4 slides on my last 2 years of experience talking about DARPA and how it got itself into biology, and we have experts here that have been involved with this even longer than I have. So give you a flavor of what I had been doing there and the vision at DARPA. One slide I'll talk about BioMEMS, at least my take on BioMEMS and where the future might be, at least one version of it. And the meat of the talk obviously is going to be focused on my past and current research mainly done at Lawrence Livermore National Laboratory and focused on microactuators. And then there's 3 application areas. The first one is interventional neuroradiology for mainly stroke treatment, but other tools that might result from that research will also be mentioned. And then there's a second area of application is going to be focused on microfluidics or microflow processors based on magnetohydrodynamic forces. If I have time, I'll spend time on the third area that I worked on, which is basically an optics application of MEMS and interferometry. Not quite bio, but it has implications of how actuators can be used for instrumentation, optics, and a lot of it could be applied towards bios in the future. And if I were at University of Washington, what would I do? So it'd probably be an important topic as well. Talk a little about what I think about my personal plans and goals and resulting from the vision that some of it developed from the combination of Livermore and DARPA. Okay, so what is DARPA? DARPA, most of you probably have heard, at least claims itself as a true inventor of the internet, and also has a lot of experience over the years through Semitech and other investments in photonics, for instance, VCSELs, Semitech doing the IC chips, basically being in the 80s, allowing Intel to take. I think one of the reasons we were able to take the lead in the chip business was some of DARPA's investment. But anyhow, we've been focused on large-scale, for instance, computations, algorithms, microscale chips, and Most recently, 5 or 6 years ago, we started to be involved with biology, mainly motivated by the threat of biological and chemical warfare. But I think in the last couple years, DARPA has taken a step back and looking at, we got ourselves in a rush and just got right into the bio business without looking at the fundamentals. So we actually are starting to look at the interaction between what DARPA is traditionally strong with in terms of technology development in, for instance, micro and nano scale technologies, as well as information algorithms, advanced parallel processing equipment. How do we take that and maybe help, because the length scale and the time scales are now approaching the fundamental length scales and time scales of biology, how can we take that to probe and interrogate the biological understanding of cell and molecular biology? So we're taking the physicist technologist approach, but really pushing it towards bio. But on the other hand, we figured that by doing that, we understand more about biology, understand more about the massive data processing techniques and the very complex systems issues, and we can apply that back towards the defense sector in developing better information and microtechnology. So there's a two-way stream here, and I hope in the future DARPA will continue to go in this directions, and I'll probably at the end, part of my vision part of the talk will focus a little bit on some of the future directions that I would like to be involved with as well once I leave DARPA. But there's also many obvious, let me use this, many obvious DoD applications, including combat casualty care. How do you take care of wounded soldiers in a very efficient manner, and knowing which is the most critical patient that you can make a difference with. But there's several others that I won't elaborate on. This is my current journey. I mean, my personal journey at DARPA is going down the length scale in terms of, and going down up in the, integration scale in terms of number of components per chip. So basically, we started out microfluidics. I took over in 1999 in February, and we focused a lot of development because of its early stage understanding fluidics in terms of microscale phenomena and surface separation, surface interactions. Limited us a little bit in terms of, and it's a very natural thing that we limit ourselves to a lot of components development. How do you make a microscale valve and pump? How do you prepare the surface so you don't have protein absorption, or prepare the surface so you can control that protein surface interaction? But because of that early stage, we needed to develop components separately, and much like the electronics industry did early on, like breadboards, you have components put in on a substrate and start connecting them through that motherboard or substrate. But that's the general rule there. But in terms of BioFlips, which is the program that was followed on microfluidics, we focused a lot on the integrated circuits version of microfluidics. So we focused on platforms. How can you make a platform like the CMOS process allows you to integrate many components that with multiple functionality, much more complicated, obviously, than the integrated circuits, because you're not just moving electrons. And so these length scales now started to have chips have the potential of putting hundreds of devices on the same chip. But really, to get down into the nitty-gritty, this is still like a microfactory on a chip. There are some different physics phenomena in terms of the dominant parameters, but really you're not quite getting to the molecular interactions, which is sort of as you heard in every area they're talking about nanotechnology. But in the way I interpret it, along the same roadmap, if you may, it's getting to the molecular scale control and the molecular valves and nanoscale control of transport. This is really a true marriage between science and technology. People still need to do very good microfluidics, implement maybe hybrid devices. Say you might have a protein in a artificial or synthetic cell membrane, and allow you to control with transducers interface the molecular transport, filtering, and very specific allowance of specific molecules, either through their charge or through their sizes, to let them come through and monitor those events, much like the cell does. So we're taking the cell, very complex thing, taking its individual components apart, maybe using the cell membrane as a platform that is much like a platform like silicon. When you don't dope it, silicon is very good insulator, and doesn't allow current to go through. But once you dope it, you have these transistors that are phenomenal in terms of control at that scale. So the same thing could happen here. That's why Microsystems Technology Office, the office that I'm in, is trying to get into using that as the metaphor to control that scale of biology and also interface to biology. And we are also working with a lot of the science offices and technologies. Okay, and the last slide for the introduction of the not meaty part is BioMEMS. Again, my take again is when there's two sides of medicine. One, of course, diagnostics and therapy, and one is more in vitro and one is more in vivo, working with the body. There's a nice thing for most of us to realize that there's a potential for a lot of cash flow if you do it right. But Nexus from Europe has at least estimated, one estimate is at 10 billion by 2002. Three main areas: in vitro diagnostics, hearing aids, and drug delivery. So those are still important areas and a lot of potential to expand upon. But in terms of MEMS, it's the same thing. I mean, you go to it's the sequential fragmented medical paradigm that you have. You go to a doctor, he takes tissue or samples from you, urine or blood or other kinds of tissue samples, allowing him to send it to a lab, eventually come back, and he gives you a prescription for a treatment. So it's a really sequential process. Not a lot of so the technologies to do these things are usually separate, because there's a lot of different constraints in each, and less constraints, of course, in vitro. Whereas MEMS is the same thing. There's been tools to do, for instance, drug delivery is a big thing, skin patches, and recently there's a gluco watch, just approved by FDA, having just micro devices interact with the body. But we deal with the host anatomy, and there's the other side of it, of course, the genomics and proteomics and the drug discovery and drug testing allows us to deal with samples outside of the host, but it's derived from the host. Just my take again on the future is hopefully MEMS and other integration technologies and a combination of different expertise again will allow us to really have parallel processing, having a chip side by side, have the capability to do not only early detection as well as real-time monitoring and feedback to the body and the living systems, and allow for potential for integration of detection, diagnostics, and treatment in one system. So next, again, I mentioned I'm going to talk about my past research, and some of them are still ongoing at a very small scale, obviously. Microactuators, I'll give an overview. I focused more on microactuators. There's, of course, a big field in sensors, biosensors, there's lots of publications since the early 80s. But I've pretty much taken the road of microactuators with the mechanical engineering background. Then the two application areas, as I just mentioned earlier. Oh, before getting in, I will talk just a little bit, just introduce myself a little bit more. Is the two main things I was involved in at Livermore was a very good opportunity for me at the time when I graduated, and that is they just built a clean room. They just built this new center for microtechnology, and this is a typical clean room. Later, as a group leader, at least I was involved with managing and maintenance issues and personnel issues, technician issues, precious metals issues. But that's, please give you a background of I had some background in terms of managing the clean room, and I look forward to seeing more of the UW facilities. On the other hand, I was working a lot with the new effort at the lab, and was the medical technology program, and focused on using technology at the lab, and my specific is using MEMS towards key national level healthcare problems, basically the three leading causes of death. Okay, so microactuators. Obviously, microactuators, like most everything else, is not a one-size-fits-all solution. So early on at the lab, we decided, I mean, there was no microactuators at the time. We decided to develop a series of them. But the main thing is how do you design them? What are the constraints? What are considerations for design? Obviously, when you have an automobile, you have torque versus the frequency limitations and plots. And so we felt that this is at least one way of characterizing actuators, microactuators specifically. Work per volume, basically how much work can you output with the unit weight or unit volume, and then how fast can it respond. And so as you can see, like shape memory alloy ranked pretty high in the amount of force and energy output that you can make, but not very high, about 100 hertz in cycling frequency. Then there are materials that really have high cycling frequencies, like the piezos, but they're okay in terms of work per volume, but not the highest. So it depends on what you need to use it for. And I'll get into a few actuators just to illustrate the things that we developed. And obviously there's other constraints. Fabrication compatibility is one key one. Even if you have the right kinds of, it's in the right part of the chart, it may be hard to integrate. It may be not compatible with the facilities. And then biocompatibility, obviously, for medical applications. Just one actuator I mentioned in my early work is an actuator basically just to take that chart there in consideration. Using a resonator, you can generate high frequency actuation. However, it's only repetitive motion. You don't have continuous motion. If you combine it, if you transform or convert this energy into a continuous motion by connecting it to a rotor or a linear slider, you allow yourself to transform this or convert this repetitive energy or motion into a continuous motion. And then that allows, in this case, this is kind of a neat thing. I like to use this just for illustration. But if you have these two resonators, these are comb drive and spring resonators, and they move in phase, this bar will go up and down and move this rotor in the clockwise direction. If they're out of phase, this rotor here, tip, will basically slap left and right and turn this in the counterclockwise direction. So this is just a close-up SEM of it. With this, you can control very, very fine, because of the high frequency, very fine control of motion, and was used later on for aligning optical components on a chip. For instance, a laser diode to a optical fiber. And then there's a lot of impact dynamics obviously involved with this. We looked into the wear and the dynamics, the nonlinear dynamics for this design, but it allows you to, like a jackhammer, just robustly adapt to the resistance that it's facing. So in the early motors, they had electrostatic motors that got caught up with the static particles or the restriction effects, but this was able to overcome that. Like a jackhammer, when it hits a something, the frequency will match the impedance. And then a couple more actuators that I just mentioned. We actually focus a lot on bimetallic or bimorphic actuators. And the reason being, I mean, these are very, very simple theories. I mean, basically you just take thin films that, I mean, a lot of it is developed really extensively by the semiconductor industry. You can develop right kinds of thicknesses and and have different material properties. DuPont's and other chemical companies allow you to design in very many parameters. Mainly the two parameters are the coefficient of thermal expansion and the stiffnesses, the Young's modulus. Once you put these thin films down and you have a annealing or a temperature difference, allows you to generate a thermal residual stress, and that will curl up this so-called bimorph cantilever. And this is just some work we did earlier, just using different lengths. You allow you to have different kind of initial curvatures. If you're using polymers, even more so, you can get the stress. Okay, here's a stress relationship between the difference in CTE as well as the difference in temperature. But you can calculate with Timoshenko's formula the radius of curvature. Again, back to the polymers. If you do polymers, again, you can get the insulation you need for most of the fluidics applications, the good seals, and good insulation, so you can embed in electrical components that are shielded from each other. One application of using these thermal bimorphs that gives you large deflections with polymers is a microvalve. You could put, again, embed inside the first layer a metallic layer, which gives you a well-defined gap between that electrode layer and the top of the substrate, which are both conductive, and you apply a bias voltage, allows you to zip up it with a critical voltage that overcomes the mechanical stiffness to zip up the normally open valve, and the pressure will be held off with the critical voltage calculation, you can get pressure hold off here. And we were up to about, it's not very large forces, but we're up to about 3 to 5 psi at maximum, because we found that the flexibility actually was not a good thing when you have two materials that were adjacent to each other in terms of the softness. If you start deforming the membrane around the periphery here, then you allow initial crack to generate, and much like fracture mechanism, it will just propagate that crack. So the hold-off pressure was not as much as we expected. On the other hand, it was used for connecting from Livermore was developing this PCR micro heat chamber, and then and was trying to connect that to the electrophoresis. Early work in integration of different biological instrumentation, and so we didn't need a lot of pressure hold-off. All we need is open up and have a controlled droplet push the already cycled thermal cycler into the DNA and amplified DNA into the electrophoresis chamber. We tried a few times. If you were going to ask, it did not make the final human genome project, but it was one of the efforts. Another way to use this thermal bimorph is using the different materials. In this case, a muscle-like, mimicking the muscle motion, is the conductive polymers. And the way it works is basically the polymer backbones will be doped with certain kinds of ions, and these ions can be driven in and out through with a voltage bias. And it's really low voltage, 2 milliwatts. I can take this conductive polymer that's about 360 degrees in a circular configuration and completely open it with relatively large actuation stresses. So it's another way. It's basically a chemo-mechanical actuator that could be used. Okay, those are just a few examples of actuators. You can just talk about two application examples. The first one is as you know, the catheter technology is being is really becoming more of a common thing in cardiology, for instance. Angioplasty or stent deployment has become very, very a normal procedure, and it's really a nice procedure because people go in and out and very, very little in hospital inpatient time. So it's pretty much outpatient in some cases. But we were trying to extend it to the brain because we made so much progress over the last 20 years in the heart. But the same technology, there's no reason why at least a lot of it can't be applied towards the brain. So when I joined the lab in 1992, that was my first project, was to develop the microactuator to release coils into brain aneurysms. So basically, a brain aneurysm, as most of you know, it's a bulging and weakening vessel portion that will continue to balloon until it bursts and cause a hemorrhagic stroke, which is a critical thing for healthcare. There's not much to do about stroke. I mean, it either happens or doesn't, and you just take care of the results. And take care of the consequences. But the key to do this is, again, to release this coil. It's a one dimension. Of course, it's a threaded wire, but once it comes into the aneurysm, it starts to coil up again. It becomes a three-dimensional ball-like thing that fills up this chamber. And because it's coated with embolic material, which means blood comes in and coagulates, and eventually closes up this aneurysm neck, and allows blood flow to continue to return to normal, and that will eliminate the ballooning effect. What they're doing now, state of the art now, is basically using electrolytically dissolving these junctions to release these coils into the aneurysms. But that has a lot of problems. Basically, it's not as reliable because it's a chemical effect, and it actually takes 4 or 5 minutes for each coil, which sometimes you need 10 or 20 coils. So the key was to develop something that's like a gripper or some kind of mechanical release mechanism that instantly can release a coil, end of the coil, into the brain. However, when you're going through meandering through the brain and the bends and turns, you cannot let go prematurely, especially if you're not completely in the right position. If you prematurely deploy it, then you might cause a stroke on its own. So those are the problems we're faced with. You need very good, strong grabbing force before it's deployed, but you also need to really have a complete let go when it's in the right position. And the physicians that we're working with, they're from UC San Francisco. Basically, what they want is something very easy to measure. They want to pull it and see if they can get up to 30 grams of pull-off force. As a MEMS early, a young graduate, the first thing, of course, the first project that I tried to solve this was obviously with a MEMS solution, taking the hammer that I know of to hit it. But so anyways, I'll quickly go through it, but tell you why this was a good experience, but on the other hand, it was a learning experience. Basically, we're looking for something pretty rigid, as I mentioned, it has to be rigid, three-dimensional, going through something that's less than a millimeter in terms of the microcapacitors at that time. And so we need a really what they call now high aspect ratio micromachining. At that time, there was no deep RIE. So what we ended up doing was we can etch deep channels with wet etching because of the crystalline plane, anisotropic crystalline planes, but we can only etch it in one direction with the 110 wafers, and that allows you to have 111 planes in the vertical direction in a certain crystalline plane. So you can etch that. In this case, it's these vertical lines, but you can't etch the other direction so with the other direction, the only way we could figure out was using precision saws, working very, very hard to get very good control of how deep you saw into the wafer. So this is this direction. The other thing you needed to do is we wanted a bulk micromachining effect and batch fabrication. So we wanted to be able to make many, many grippers at the same time. And if you want to bond these two wafers together, of course, you need to bond only for instance, the base portion, so that the other open portion can open up. So we did a lot of process, very complex, and we had to, of course, how do you generate the force? We looked into that chart again, looking at the shape memory alloy as the highest work to volume ratio. We didn't need very, very fast response times, so that was ideal for this case. But then that's a lot of development because of the high stress and high temperature we were hit with. But this is what we set out to do. And the technologies that we had in several papers we had to go through to get to this point. This just shows one part of the development, how we did the selective eutectic bonding, basically putting, and this is top wafer here, but we put the gold on, and again, a lot of control needs here because the eutectic point will change drastically if you don't have the right, if you have oxygen basically exposed to it, it generates, the eutectic point shifts dramatically, so you don't get the true eutectic effect. But here shows the results. Basically, infrared imaging shows you pretty uniform in IR transmission, allowing us to believe that you have that right eutectic bonding. And further verifying by looking at the SEM at the cross-section, it's a very sharp contrast of where it's bonding and not. But the final test, of course, is the pull-off force, where you found that the bottom line is the pull-off force, the bonding strength was higher than the fracture strength of silicon, which is the best you can get. And so we bonded here, but we needed to open it up and close it. And this will be this is a lot of explanation to do, but I worked with the colleague Peter Krulovich, and he was the material scientist to develop the magnetron sputtering technique for this nickel titanium copper. And just to make a long story short, the copper addition allows you to really focus in on a certain transformation temperature range, and also gives you relatively narrow hysteresis, so you have relatively fast response times. And the other critical points are you still maintain the high stresses that you have with nickel titanium. So the copper addition actually is more for manufacturability, because once you don't have copper, nickel titanium shifts by a few percent. You shift the transformation temperature by tens of degrees. So it's hard to really control that. Again, this is not a thermal bimorph effect, which the earlier one that I mentioned was. It's a phase transformation effect. So you put the shape memory alloys on top of these cantilevers at high temperature, to 500 degrees actually, and then you anneal it to crystallize it, but have a shape memory effect. As it goes down, the thermal stress actually allows it to curl up until you hit what they call martensitic phase, where it starts to become soft, like basically it allows it to have twin boundaries to have large strains. And then the silicon eventually, the stiffness of the silicon allows it to bend backwards and become flat again. So that's how basically it works. When you heat it up, it remembers its original shape, which basically is a stress state, and then it pulls in and opens up the gripper. Just this SEM showing the gripper showing basically we use this to do a lot of the power consumption tests and how we deposit shape memory alloy with this shadowing effect. But I went through a lot of that, and we actually got this on like the cover of some magazines, and a lot of people were interested in this. And we actually, but at the end, the doctors just could not allow for two things. First of all, the torqueability was not quite there. The pull-off strength was okay. But the main thing, which surprised us, they knew early on that these will be, there would be pretty sharp edges. But the sharp corners was a big concern for them because of how it looks. It just feels that you're going to scrape through your blood vessels, and nobody wanted that. So this was pretty much dumped, but it attracted enough interest to be able to do this. We had another trick under our sleeves, I guess, at that time, which was not quite a MEMS solution. So it tells you that the main thing is to solve the problem at hand. We used another shape memory phase transformation material, which is a polymer instead. This polymer here has a different kind of phase transformation. Actually, at the high temperature phase, which is the rubbery region, it's actually only megapascal. It's very, very soft. Where at the glassy region, the lower temperature is very stiff. It's like Kapton. It's really, really stiff. So it's about 400 times change in the stiffness. But the memory stage again is the high temperature rubbery region. So it's opposite of shape memory alloys. And so how we designed this again is using a fiber optic instead of using electrical current, and putting in the end of the coil here. If you put it in, you heat it up, becomes very soft. Again, 400 times softer. You can apply uniform pressure to conform this material around it. And once you cool it down, it locks in. Because remember the memory position is the high temperature position. So once you, and then this where a lot of calculation went in for getting uniform heating of this material because you don't want hot spots. Once you heat it up, then it remembers its original shape, which is the open shape, and it instantly opens up. So this happens to be the solution to this problem in a sense. And then there's further advantage using fiber optics. You can look at the reflection intensity changes to look at, so when it's intact, for instance, the coil is butt against the fiber optic, you have a high reflection efficiency. But when you're at the opening coil release, which is kind of critical for a feedback mechanism, knowing when it comes back, when it's actually there's a gap is increasing and you know that it's released, because the worst thing is it's not completely released and you pull it back halfway and then it releases, as I mentioned earlier. And this is the IR imaging showing that we actually got fairly uniform temperature, at most 2 degrees. It was 1.1. Basically have about 1.2 degrees total difference in the temperature between the edge of the fiber optic and the end of the shape memory polymer. So we had a feedback mechanism, and this actually went into animal trials and human trials and was pretty successful with the company, which started with one person. It was about 40 people when I left for DARPA. So the next area I want to talk about is a microfluidics application. We started working on this Magnetohydrodynamic microflow processors. And it's for the in vitro side of my talk here. But the bottom line is we wanted to do something that's like the integrated circuits. You can tune in individual local control of the flow and the chemistry. And what we used was this thing. I mean, as you know, in electrical engineering or physics, basically, there's a Lorentz force when you have an electrical field and magnetic field that are perpendicular to each other. Like all our motors work that way. Then you have a Lorentz force in the perpendicular or normal to these two fields. And so if you put it in the channel configuration, you think about a wafer as a two-dimensional thing. If you have the magnetic field that is a uniform field that's across the whole wafer, and you apply a current in the wafer dimension, then direction, then you can allow for a force to be pushed in the third dimension, and that could be pretty much match what a microfluidic channel would look like. So if you have B field normal to a E field, then you produce the Lorentz force. Problem for a direct DC fields arrangement, the problem is that you easily generate electrolysis, because about 1.2 volts, theoretically, you start having bubbles generated in most ionic water concentrations. And so AC current actually allows you to bypass. If you reverse and synchronize these two fields fast enough, then you don't allow enough time for the bubbles to create the critical threshold. So there's a frequency limitation, of course. But this is just this showing the forces that you can generate, resulting into a pressure that's driven inside the channel. And you can allow, again, if you look at this formula, there is a phase in there between the two fields. If you adjust the phase, you can adjust the magnitude of your pumping force, but you can also adjust the amplitude of the electrical or magnetic field, and get that's the flow rate. And so the advantage, of course, here is you have no moving parts. There's no flapping membrane that needs to push it for a pulsatile flow. And of course, but one limitation obviously is you cannot do non-conducting fluids. But most biological samples have some charge in there, or you can have enough separation so that you can generate that force. And remember, the force only is generated in this electrode cross-section area. So once you're beyond that area, then you start generating pressure-driven flow, which has some advantages. And actually, I'll get that. So this is a simple configuration of what we did, basically anodic bonding of three of the glass and silicon and glass sandwich layout, and then you have just electrodes in between. This is the first generation of the pump. Basically, these are the electrodes that are at both sides of the channel, here and here. You apply that, and the magnetic, again, can be pretty small. I mean, this was not strong enough, but it was enough to generate the force we were looking for. There's a quarter there that looks at this chip particular compared to the size of a quarter, and this is a total package. A very simple setup. The two fields requiring synchronization between and the phase control between them, allowing you to generate the electrical field and electromagnetic field, so that you have a fluidic force generated. Again, I mentioned about the bubbling or the electrolysis issue. It's a function between. We did not do a lot of theory, which I think is something that should be done in terms of theoretical work. But we did some experimental work looking at the different concentrations. Obviously, it's a function of concentration as well as frequency of the force fields. And higher the frequency allows you to get higher current amplitude, which is important to give enough head or pumping force. And then the higher concentration gives you more ions to work with, so you generate more force. And we use basically the image capturing technique to judge the flow speed and therefore derive the flow rate. ABC, if you look at here, this is outside of the electrode or the pump area. So you can see ABC started out here. After less than a second, B actually bypasses C or passes C because the initial stage where they were in the pressure velocity profile. I mean, the C was probably closer to the edge of the channels. Let me see if I missed something. Okay. Oh, well, the flow rate is not very strong for this particular magnet, but we got about 20 microliters with this kind of dimension. Again, it's very nice because you can change, as I mentioned, you can change the phase. You can change the phase 180 degrees and allow you to reverse the flow. So we instantly reverse the flow because of the low Reynolds number. Go back and forth instantly, and so the amplitude can be controlled as well with the phase. Just show some flow measurement results matching the theoretical numerical calculations that we had. And the reason for this big envelope here is because we had trouble measuring the magnetic field. So we basically looked at the top of the field, top of the wafer, and the bottom of the field, and look at the envelope containing the flow velocities. And so again, controlling the amplitude by phase or by amplitude of the electromagnet magnet. Okay, this is a table that just illustrates all different fluids that we looked at, concentrations. We actually had one that was pH close to 7, and it was still giving us relatively smooth operation and okay flow velocity, not quite the same as the higher concentrations. And we also pushed DNA around in the channel as well. Didn't do much experiments with it, but we did push it. We did put it back into the PCR chambers and looked at that it was still functioning DNA, is what I'm trying to say. And so once you do this, that's the fundamental component. You can do a lot of things. This is just one example logic a switch, right? So if you apply, again, there's 3 arms. One is arm 1, arm 2, and arm 3 for a switch. And if you apply just the first pump, P1, with the electrical field, then you start drawing fluids from both arms. By applying a counter pressure in one of the arms, then you can pretty much use it with the force flow sensor. You can pretty much balance out all the flow that comes in from this arm, and allow it to come only from arm 1 to arm 3 if you're going to switch back and forth. And the same thing, of course, if you apply pressure from the counter pressure on the top here, you allow you to switch back and forth and instantly again from arm 1, arm 2 into arm 3. And this is not the best example because we ideally need the dimensions to be much smaller, because here you have a swirling motion. Basically, viscosity allows it to swirl around. So the switching effect is not as good because you have mixing actually at the junction. But it shows you the phenomena. If you look at the large picture, it will actually go from moving in this top circle into the bottom instantly with the changing of those two of the switch. This just shows, it's actually not the best result, but it shows we are switching until a certain phase, 45 degrees in this case, that allow us to have 0 velocity in the third arm, in arm 2. Is that true? Yeah, arm 2. But I do intend to go further in this, but for today's purpose, these are some examples of how you can use this. This is start thinking scaling up like the integrated circuits did. You can start putting these electrodes anywhere on the wafer or on the chip with once you have an electromagnetic source, of course. Then you can put pretty complex circuits onto the same chip and wafer. You can reverse the phenomena from a flow pump into a flow meter, because all you need is to measure the voltage drop when you have a flow going through there, and the magnetic field in particular to the wafer. Again, you generate a voltage drop, and you can back out the flow rate. You can also, once you have the flow rate, you can plug it into the normal pressure-driven flow formula, and then it can back out the viscosity. So again, this is a platform that allows multifunctionality. You can do sensing and actuation. Lastly, one more application. We actually applied it. Now it's currently ongoing program using this pump to do an integrated biological sample preparation device. And this is a simple configuration of it, basically taking this buffer solution and the spores that are in lysine solution, so the DNA is starting to be extracted out. But the main thing is using the pump to pump the buffer and control basically control a sample buffer stream with a original sample stream, and then with a DNA extractor pulling only the charged particles across that flow interface. Then you allow the DNA to be pushed to the detection system, while the rest goes to a waste. So that's what's one of the simple sample preparation jobs that are being done at Livermore. Other things that we propose, of course, are modular switches, just having these modular things that we just connect to each other and allow you to switch. I want to send a sample to a flow cytometer or a PCR chamber. You can also do combinatorial chemistry now, starting to have really good control of the flow rate, allowing you to say different concentration ratios at these channels for different, again, combinatorial chemistry or drug discovery purposes. Let's see, do we have time? I guess we would like to leave some time for discussion. So I'll just simply talk. This is just I did quite a few optical MEMS projects as well. This one was motivated by this interferometry that was actually state of the art in terms of being able to take large scale optics, reflective optics like this one here, and use this, and I won't go into the details, but use this phase shifter here that will allow you to get down to angstrom level or sub-angstrom level of metrology of this large scale optics. It was important for the extreme ultraviolet lithography program. So what was the key here was taking this PZT actuator phase shifter that needs to control 1 or 2 nanometers to get that kind of metrology, but it was a 30-pound 3PZT, which is need no feedback control. It's about 9-inch by 9-inch, but all you're moving is about a nanometer or 2, actually micron stepping motion. So it was really an ideal application for MEMS. I'll just use this last. There's quite a bit of slides here, but I'll just focus on this one here. So if you think about it, we just use a very simple electrostatic comb drive configuration, and it's very symmetric. We're not driving it in a lateral direction. We're actually driving it in a vertical direction using what they call the levitation force. When there's an asymmetric electrical field, you to have a resultant vertical levitation force. And if you balance that with basically a spring counter force, then you can control the direction. And how do you do the feedback? You have this large area here for you to do a capacitive feedback control. So we're taking this huge thing here and kind of shrinking it down to only a millimeter or a centimeter in terms of total package for doing that phase shifter. And that would be kind of the key is if EUV lithography becomes, I mean, they're still thinking that's going to be the winner based on many other reasons other than technical as well. But then they need these metrology tools to be all around so people can measure fine optics very, very accurately. So this is the MEMS version of doing that. Based on time, I won't go into the details, but just to give you the results, this is the original configuration. Of course, you have the squeeze film damping effect in one direction when the mirror is released and it comes down and air needs to be pushed out of the gap there. It has a very, very low response time. But when you're pulling it up, actually you get very faster response time. But the bottom line is it's a nonlinear effect. Using that capacitive feedback, we were able to get accuracy down to less than 1%, and that kind of translates into 2 to 3 nanometers of accuracy control, and that was very critical for that interferometer. And that's the package and the final mirror, and with the on the surface mounted package. Okay, so the last part is what kinds of future things. I mean, we actually have, again, I have quite a few patents in this area of catheter procedures, micro devices for steering. We're looking at ways to do wireless control instead of having to use the optical fiber or the electrical wires to control. We have ways to do local drug dosage control. So what I was thinking here, again, with these tools that I have some initial concepts with, cauterizer to do cutting or removing of clots or of plaque. But what we're trying to do is have a suite of tools that looks like the laparoscopic surgery today, being able to do, if you do laparoscopy with an abdominal, you can look in there with a fibroscope, you can use you have cutters, you have ways to tie knots and so on. But if MEMS can push the state of the art in terms of pushing catheter tools to have that kind of complexity or sophistication in doing things inside these at least the axis, I think will open up a whole new area of different applications for treating and for diagnostics. The second area is, again, expanding upon the fluidics to really push further in the integration scale-up approach and working with people that have the skills in the bio and chemistry and the surface preparation, which I know UW is very strong with. Look into, again, the nanoscale components, making lipid membranes, making maybe liposomes for drug delivery, but controlling at that scale, using fluidics to deliver and manufacture these components and then monitor the interaction between them. But again, there's some specific examples that I would be happy to talk to you about. But this is the hybrid molecular dynamic network that kind of is hopefully is part of the proteomics and the next functional genomics direction that everybody is pushing, but with more mechanical approach, I guess. Here I know, I didn't finish this, sorry. I was on the plane yesterday. Polymer microfabrication, and of course this is, I mean, Paul has done a great job here in pushing and specific kinds of polymers can be used for more biocompatible devices. But I would like wherever to build up. I mean, the whole range of it. I think this is what you need. And you need to also integrate it with maybe the high aspect ratio MEMS and the silicon micromachining. But I think the future is you have to get into the polymers, and so I would intend to continue that with collaborations with many people. And then I want to look into taking shape memory polymer, which I have had no handle on, and to put it onto the MEMS scale. And I think there's a lot of devices that I could work on. This is sort of taking, maybe you can say stolen now that I'm leaving DARPA, but number of transducers versus number of, it's one of the roadmaps that you can do. Remember, we can do these computers with a lot of integration, scale up, not a lot of components on one chip. Biological components, the DNA arrays are getting to be very, very large scale, but there's not much interface between the control and monitoring of these. You either do a hit or miss thing here, or you do a control with only electrons here. So can we do a control with the large-scale biological molecules to have chips that can really perform not only what this as a cell level, but basically a design cell functionality. I think I'll use this last slide to sum up. Basically, there's a lot of opportunities. I think, as I mentioned, in vivo and in vitro can be combined to make a big difference in the healthcare sector. You can have devices that work with the body. At the same time, the device will have the nano and molecular scale interfaces that allow you to have direct control and monitoring of molecular events that affect eventually up to the tissue, up the organ, up to the complete health level of the human being. And that's because of the length scale, again, I mentioned, and the micro components that you can do control and do scale up with. But I hope we'll have the opportunity to discuss more about my ideas with you with some questions. And thanks a lot for your attention.
Thank you.